Word. Okay. I realized that in this course, it was from last year's final exam. I realized a lot of students learn all the tools, but they don't know how to use the tools. So I want to show you a few scenarios. Pretending you are the computational biologist and your advisor is the experimental biologist generating the data. It's like, oh, great, I sent you to take this course. Now you can help me analyze data. Or pretending you are the student in a collaboration where you are with a computational group and you're talking with another experimental group and they have some data and you guys are kind of talking together to analyze it together. So let's look at a few cases. Okay, so you observe that there's, your collaborator comes, they have a drug, and they see that when they treat the cell with a drug, the cell is dying or like growing slowly. There's some, effect on the cell growth. And so they did a microarray experiment on the cancer cell line. So with the drug and without the drug, and they look at the time course. So 0 hour is like basically nothing has been treated yet. You can imagine 0 is just no treatment. And then 1 hour, 5 hours, 24 hours over time in 3 replicates. It's done in different time. And they have, in this case, 4 time point, 3 replicates, and with and without treatment. There's a lot of samples here. And they come to you, oh, I need to publish. Can you help me with analyzing the data? What do you do? What do you use first? And by the way, we will have similar type of questions in the final exam. So we wanted you to start thinking about this. What do you do when you have this? First of all, what files do you get here? You have cell files, right? What do you do first? You just use RMA, right? You normalize, you do medium polish, you get your gene expression index. Then what? Sorry, I already told you. You use differential expression. How do you do the differential expression? Claire. Actually, if you run Lima, you can first do things like, you say these are all different conditions and just give me any genes that are differentially expressed at all, because the baseline is, you remove kind of the baseline. Everything else, because even without the drug, I guess assuming 0 hour is the without drug treatment in some sense, right? After the drug treatment, you can use the 0. I wouldn't think that this group will bother without drug treatment to grow the cell for all this time to do the experiment. So the 0, without the drug would be kind of like 0 hour. And you can use your 0 hour as the baseline. You look at 1 hour, 5 hour, and 24 hours, all comparing with your 0, right? You can run Lima for that. And then what do you do next? Yeah, exactly. Supposedly, you don't have a lot of differential genes for some reason. You just want to check. You do any kind of a clustering analysis. What do you do with clustering? Yeah, so for example, if you just need to cluster the samples, and if the sample is in the order of 10 or 20, it's not worth doing a PCA for, it's like, well, you can try it. It won't take too much time, but imagine for this number of cells, you can just use a hierarchical clustering, or you can use PCA to look at all the cells, all the replicates, all the conditions together, and see whether the cell really cluster by the conditions, drug treatment, no treatment, or 1 hour, 5 hour, 24 hours. And if what you get is, replicate 1, replicate 2, replicate 3, or if you don't get any differentially expressed genes, that's one thing you want to really check. Okay? So yeah, you do some sample cluster to check for batch effect. If you do have batch effect, then what? You run something? You run combat. Yeah, you remove the batch effect. You rerun your differential expression. Yes? Yes, you should do that. Yeah, these are all good practices, right? Right. You just get the expression index. You can already cluster the samples, check for batch effect, remove the batch effect, calculate the differential expression. Then what? Sam. You are David. Yes, you do. Well, okay. The first thing you might want to try is to cluster the genes. Because in this case, because it's a time course, you might see some genes always going up and another gene go up and then come down, or some genes going down, right? Like, so you should separate those classes. So you cluster the genes based on the conditions. Each cluster, I mean, for drug treatment, you probably have a few hundred genes, or yeah. Yeah, question.
So to be able to resolve like those up and down, or up usually, then you can't use 0 as the baseline for all. Like then you have to use like the 1 hour as the baseline for 5.
Oh, so in this case is you only look at the genes that show differential expression in any condition. We include all of those genes in your whatever k-means clustering. But when you do clustering, you don't use differential expression. You use the expression index from RMA directly for 0 hour, 1 hour, 5 hour, and 25 hours. And you look at all of those genes to do the clustering, okay? So you cluster your genes, you will see this is your baseline, and then it becomes like red, even more red, and yeah, so something like that. You cluster your genes, and then for each cluster, you try different clustering. Depending on how many conditions, right? The more conditions you have, the more cluster you might have. But for this number of conditions, yeah, probably 5 or 7 clusters would be enough. Once you get the clusters, you run David on each gene cluster to see, okay, this group is doing this, that group is doing that. What else can you do? You kind of want to know what this drug is doing, right? Potentially, you want to find the drug mechanism. And actually, one way to do that is you run gene site enrichment analysis. And there are also other tools in the public. We didn't talk about this, but is there some cancer? Supposedly, this is a cancer drug or whatever. You based on gene site enrichment, it will show what, like some other papers generated this gene list 1 year because that group is the differential gene between cancer and normal of say breast cancer, and your gene or your drug, when you treat the cell with the drug, those differential genes are the same genes that are get differentially expressed in cancer. Wow, that's really great, right? So based on sometimes the gene ontology, like David can tell you some mechanism. It's related to the knockdown of one gene. Then you say, maybe my drug is inhibiting that gene. Or if tell you the differential gene after this drug is similar to some disease or to metastasis or some other things, it will give you a lot of actually ideas for what those data or what other data are relevant. And so based on that gene set, you might be able to go to the original publication to pull out that original data to see, what is that study? What did they find about that condition? What type of experiment they did in that paper? Okay, so that will give you some idea what other public data are relevant. So that's the, yes, sorry.
Does it make sense to try to remove effects of other machine accounting variables like patient's ethnicity or something? Can you run batch like combat again after you've taken out the effects?
Yeah, depending on what you have. I would think this experiment, because it's fairly simple, it might be okay. If you have a much more complicated one, you might want to run SVA to find unknown hidden batches, right? But by the way, in reality, if you have a collaborator that come to you to do a microarray experiment, you should just say, go back and do this in the RNA-seq. Yeah. But if they already have the data, well, maybe you help them analyze. But really, you should recommend them to do RNA-seq in the future. Okay, scenario 2. To check the effect of inhibiting gene X. So this one you do RNA-seq on 2 breast cancer cell lines. So like supposedly this gene is very important in breast cancer, and we want to see how it performs in the real tumors. But it's hard to inhibit a gene in the tumor. Therefore, we use a cell line as a proxy, right? You treat the tumor, because cell lines are not necessarily real breast cancer, but you want to, you get some idea. So there's cell line A and there's cell line B, and you do the you use knockdown using siRNA. You have 2 siRNA N and M, and you treat the cell with like 0 hour and 2 days. I mean, 0 is like no treatment. Say two days is because siRNA might take some time to kick in. That gene then get knocked down. You do 2 replicates. Now your collaborator comes to you. I want to see what my gene is doing in breast cancer. What do you do? First thing, what file do you get? FASTQ, yes. Then what do you do? FASTQ-C, then what do you do? Yes, you do mapping. Depending on how much your collaborator wants to know the detail, if you want to do a quick and dirty, you do salmon, right? If you want to do more, you can do star, you can do like more fancy stuff. Yeah, and then after that, you get an expression index. Say, so if you run star, actually you need to get the like RSAM to get expression level TPM. But if you do salmon, you will get TPM already. What do you do next? You want to cluster your samples similarly, right? Because supposedly these are breast cancer. You're treating the cell with the same treatment of knocking out or knocking down a gene. You should see, hopefully, without the 0 hour and 2 days for both cell line, basically without the knockdown and with the knockdown to be in 2 distinct clusters. And maybe the replicates should be together, and also the different cells may be slightly different, right? So you want to see that. If you don't see that, maybe you want to run some combat or whatever to get rid of this, right? To visualize the result you can try PCA, hierarchical clustering or whatever. You remove the batch effect with combat. I would say if you don't see strong batch effect, there's no need to remove, because combat sometimes can overcorrect things. So just double check ahead of time. I would say right now for batch effect removal, there isn't an ideal algorithm. Combat is still considered the best way, but it's still, there are still properties that we don't know well enough yet. And then what do you do next? You want to do differential expression. How do you do differential expression in this case? Sam.
Into your model of batches instead of, because you have to use your regular.
Yeah, but combat work, you can, actually I would say in this case, if you use combat, probably you will see it to first correct just the replicate. In this situation, what you might want to do in terms of differential expression, I would think is you do the each cell line, each siRNA, each 2 day compared to the 0 day. Another one 2 day compared to the 0 day. So siRNA N, you do 2 day compared to 0 day. siRNA M, you compare 2 day compared to 0 day. Okay, so you do that. Then you might want to run some clustering analysis to see whether the genes are consistent, because siRNA might have some off-target effect. Your knockdown is not successful, right? And there are also definitely some cell-wise differences, because cell A is one cell line, cell B is a different cell line. There would be some similarity, but still it could be weak. So what do you do for that differential expression? You can run DESeq too, right? Yeah. For the more complicated design, you can also try Lima-Voom. It will probably estimate from even more all the CAD samples together. Kind of a more, in this case, more like factorial design. It will try to get you that. And you want to cluster your genes. Really look at how they behave. Yes, Jet.
So would you so say for siRNA and when you do differential expression for that condition, what are you expecting to see? You're hoping to see no differential expression.
You are hoping to see, you don't want to compare N with M. The key is to compare when this gene is active, when this gene is inhibited. And so ideally, you want the N and M to give you a compare or similar differential genes when they are compared to the 0 day, right? That's the idea. But before you do that, maybe you should try it to see, because in reality, I'll be surprised if you see them completely consistent. You will definitely see some differences. And so it would probably be better to run the siRNA N and siRNA M separately. And then after you get the differential gene, then draw on the heat map to see, oh, are we getting a consistent result? And how do they work on the cell A and the cell B? And then you want to get the really consistent ones, because assuming that there's some off-target effect, the really consistent ones between the cells, between the siRNAs, that's the one that you want to more probably go downstream onto the gene ontology analysis, right? You can then do David or something. You can also run the gene set enrichment in this case to see whether it's related. But more is, because we want to see what this effect is on gene, you might want to get some breast cancer cohort to ask, let's get some public breast cancer cases and see whether those genes that are differentially expressed after the knockdown are correlated with this gene in the tumors, right? Supposedly, when you knock this gene down, these genes are differentially expressed. Then in the tumor cell, you want to know which of the things you observe in the cell line are clinically relevant in a tumor setting, right? So there, you want to see whether those genes are correlated with this gene in the real tumors. And those will give you even more robust list of what this gene is doing in the real breast cancer settings. Okay, that's another one. I have 3 more. Do you want to go over them? Well, maybe I think in this class, we probably can only do 1 more, but you can also go through them after class. Let's do the single cell, probably. Okay, single cell. We have 5 young mouse brain and 5 old mouse brain. Assuming mouse is all you bought them from the same company, it's the same genetically identical, they eat the same food, live the same lifestyle, whatever. You just want to see what's the difference between young and aging brain, right? You did RNA-seq of 5 samples here, 5 samples there. Then, okay. So what do you do? Now you get big, you run Cell Ranger, right? You already got the big matrix. You know all the cells coming from each of the mouse brain. What do you do now? Guess you haven't done homework 3 yet. We're asking you to try a lot.
Press the U buttons.
A Cell Ranger automatically do that for you. Yes.
You delete the genes that are useful.
Yes, you QC the cells and QC the genes, right? You filter out the cells that have too low and too high gene number. You also filter the genes that are too low or not zeros in all the cells. And then what? Yeah. Okay. So for this one, I guess the first thing you want to probably try is you run the dimension reduction first, right? But you do this per brain first. Just run them in a first mouse, you do this. Second mouse, you do this. Just do a dimension reduction first. Okay, put them into, then run them on the t-SNE. Yeah, try, dimension reduction, you do clustering, and you look at them on t-SNE. It's oh, great. We can see some distinct clusters. You can imagine if you do a brain, there's prefrontal cortex or whether some gray matter. Yes.
You're doing T-SNE on the cells or on the cell?
You do the T-SNE on the cells first. So right now it's each brain you do the T-SNE separately, just to see, okay, roughly they are. Let's hope that you can see the expression of each brain by, okay, you can see this brain is like maybe 10 distinct clusters. That one has 10 distinct clusters. This one have 9. Like, what happened, right? If say all the young ones you see 10, all the old ones you see 9. Oh, that's really great. Yeah, that's interesting. But also I think you will see from this, you might see that one or most of the mice you see distinct cluster, but there's one mouse there's like big blob. You don't separate them very well. Maybe you should remove that, because actually at this early stage, there are single cell samples that just don't do very well, right? So you might want to check that as well. After you visualize, you might want to annotate that as well, because each cluster, you can say, for this cluster, let's average all the cells to get an expression index. You might be able to identify some key genes there to say, that's your prefrontal cortex, that's your pituitary gland, that's your whatever, different things in the brain already, right? So you might want to do some annotation based on the marker genes in that cluster. But this is all early on. We haven't even combined the mouse together yet. Before we can do that, we really need to do the batch effect removal. You want to do the CCs. Actually, this part, I would say, I still don't know exactly this algorithm is implemented. You can try this, the CCA, and see, for example, if you were to draw all the samples together, they may or may not overlay. I would be surprised if they completely fall into the cell cluster. It could actually separate by batches. And so you might want to run this to really make sure that, all the prefrontal cortex are together, and then these are different. And only after you get that, actually, Surat, after the CCA, will give you a corrected expression matrix. You can use that then to look at the differential to say, okay, for the young brain and mouse and an old brain, are there differences in the total number of clusters they have? Or maybe for this cluster, the young brain has a lot of cells in here, but old brains have fewer cells in this. That's a big population difference. But then also you can ask, they do fall in the same cluster, but then sometimes you might see slight differences. If everything else fall together in the cluster, then that small difference in just one cluster could be most of the other cells don't change, but in this one whatever location in the brain, the old brain and young brain, there is difference. Then you can calculate differential expression using algorithm like MAST. Okay. So something like that. So I would leave you to really think about the questions. Don't just you know all the tools, but then in a real case scenario, don't know how to run them. Okay? All right.